Shall we continue the debate for another 2 hours? I've chaired a lot of symposia over the years, and the toughest thing for a chairperson is the last program, where no more than 10 or 15% of the registrants are still around. So I want to congratulate all of you for endurance and remaining for the final hour, and then the final remarks from our co-chairs. Could I have my first slide? I just have 3 to show. And I think you may have to move to the... do I have to go to the podium, or can you? Good, thank you. Very good. So this is a picture of Tito Bastianello, born in 1930, in the middle of the depression, from Venice, Italy, became one of the leading industrialists in the food industry. And have the next slide, please. And became a very wealthy and well-known and respected businessman in his country. At age 60, however, actually age 70, he was diagnosed with MDS and spent the next year or two, next slide please, trying to get 5-azacitidine, going back and forth from the US to Italy to Great Britain, and finally, when his disease progressed through AML, he got a compassionate approval to take 5-AZA, which of course didn't help him very much, and he passed away 20 years ago or so. His family, and particularly his wife, recognized what a need there was to develop better strategies for managing patients with MDS, and was kind enough to make a series of donations every year to the MDS Foundation to support the top 4 abstracts presented at the symposium with a significant check award. And over the past years, over $100,000 or euro have been contributed by the family. So we've been delighted this year, and Moshe Millman's committee, to select the top 4. The first 2, unfortunately, are on Zoom, but hopefully the Zoom network will work well. I'll introduce the first 2, and Dr. Vey will introduce the second 2. So the first is a presentation by Dr. Sushree Sahoo, who is a postdoctoral position at St. Jude's Children's Hospital. And the title of her presentation is Genetic Origins and Clonal Trajectories in Pediatric MDS. So we could have that first presentation.
Good afternoon, everyone. Before starting, I would like to thank MDS Foundation and the conference organizers for this opportunity and for choosing our work for Tito Bastianello Award. And I also apologize for not being there in person, and thank you for your understanding. For today's presentation, I will start by introducing pediatric MDS that you have heard a lot about in the past few days. So I will keep it very brief. Pediatric MDS is a rare disorder accounting for less than 5% of all childhood hematologic cancers. The patients present with hypocellular bone marrow and are classified to 2 subgroups based on the blast percentages: refractory cytopenia of childhood, the major MDS subtype, and MDS with excess blast. Loss of chromosome 7 or deletion 7q, which I will refer as monosomy 7 throughout the presentation, is the most common karyotypic anomaly, followed by trisomy 8. And germline genetic drivers constitute 1/3 of all cases, including GATA2-179L as the 2 major predispositions. Unlike adult MDS, the somatic mutational landscape of pediatric MDS remains poorly characterized. Till date, only 2 small cohort studies have been published, including our previous work, where we showed recurrent somatic mutations in ZBP1, ASXL1, RUNX1, PTPN11, and RAS genes. The majority of these mutations occurred in patients with monosomy 7, and the somatic landscape of childhood MDS differs from the adult, as mutation in genes like TET2 and DMT3A were not detected. The second follow-up study by Swartz and colleagues showed similar mutational landscape as marked by red arrows. However, both projects were based on small cohort of patients, and to date, these observations were not validated in large cohort studies. In addition to monosomy 7 being associated with increased somatic mutations, in a recent collaborative study with Evoke MDS Consortium, we showed that children with monosomy 7 have poor overall survival than normal karyotype, irrespective of the underlying germline predisposition in SAMD99L, GATA2, or wild-type cases with no known germline drivers. The fact that monosomy 7 have increased mutational burden and less favorable outcome, it will be important to define the acquired genetic spectrum of pediatric MDS. And towards that, we have set 2 objectives. Define the somatic mutational spectrum of primary childhood MDS, characterize clonal origins and trajectories of the somatic mutations with a focus on monosomy 7 karyotype. Our patient cohort constitutes 768 primary MDS cases, all subjected to targeted MDS panel with 28 genes, and 430 of these patients were whole exome sequenced to define the mutational spectrum. Representative cases with monosomy 7 and somatic mutations were subjected to bone marrow-derived single colony formic assay and single-cell DNA sequencing for assessing clonal hierarchy, trajectories, and origins. In this graph, I am showing the somatic mutations that we detected in our cohort and their association to germline predisposition on the left and karyotype on the right. A comparable mutational spectrum was observed across all germline subgroups, with few unique associations like NRAS mutations are common to wild-type cases with normal karyotype. Additionally, PTPN11 and TP53 were also mutated frequently in wild-type cases and are rare or absent in GATA2 and SAMD99L patients. Overall, the patients with monosomy 7 have high mutational load with ZBP1 and AXXL1 co-occurrence as the predominant alterations at monosomy 7 background. Due to this increased association between monosomy 7 and ZBP1 and AXXL1, next we focused on delineating this clonal architecture. Here in a representative patient with germline GATA2 mutation and monosomy 7, we harvested 182 colonies, all had monosomy 7, 164 had additional ZBP1, followed by 114 with SXL1 mutation, and 4 had Sybil mutation. This pattern of clonal evolution of monosomy 7 being the first, then acquiring ZBP1, followed by ASXL1, was observed in all 8 patients investigated. While analyzing some longitudinal samples with this clonal pattern, we found this clonal dynamics to involve unstable ASXL1 clones. Serial sequencing of the same GATA2 patient analyzed by CFC showed that before HSCT, this patient had total 4 somatic mutations: ZBP1, ASXL1 with higher VAF, ASXL1 with low VAF, and an emerging Sybil mutation, in addition to the germline GATA2, and all occurred at monosomy 7 background. Interestingly, the low VAF ASXL1 did not grow in our CFC assay. After relapse, the ZBP1 mutation presented in red grows steadily, followed by SXL1 mutation with higher allelic frequency detected before HSCT prevailed, while the other disappeared, and the Sybil mutation grows rapidly. One caveat of CFC assay is that it only represents colonies that can have the growth potential. So clones that might be facilitators of disease progression but do not grow will be missed. Hence, to understand clonal hierarchy at the native state, we used Mission Bio single cell platform. This allows to detect simultaneously detect the genomic alterations and as well as cell surface protein expression. Our panel covered whole SAMD99L genes, certain GATA2 mutations as shown in the right, 174 amplicons to detect common chromosome 7 SNVs present at 30 to 70% in general population, and also covered hotspots in 18 pediatric MDS driver genes. We added 5 antibodies for main hematopoietic lineages. However, I will skip this protein part in the interest of time. Using this panel, I will first discuss the clonal architecture of SAMD9-9L patients. Now, switching gears a bit, unlike GATA2, germline SAMD9-9L mutations are described as gain of function because these mutations do not cause loss of protein, but rather make a mutant protein which is toxic to the cells as it inhibits cell growth and increases cell death. And clonal evolution in these patients acquire genetic events which are focused in losing this toxic germline mutation by somatic genetic rescue. This involves monosomy 7, where the loss of chromosome 7 with germline mutant allele, acquiring somatic SAMD9-9L mutation to neutralize the germline toxic effect, or undergo natural gene therapy of duplicating the wild-type SAMD9-9L allele containing 7q arm called as uniparental isodisomy of 7q. And together, these observations are present in 2/3 of cases. So next, we set out to delineate these clonal trajectories. To resolve chromosome 7 karyotypic changes at single cell, we utilize 7p and 7q arm heterozygous SNPs. From bottom of the slide, cells with normal chromosome 7 maintain heterozygosity that translates to diploidy. UPD7q cells lose heterozygosity of 7q arm but still maintain the diploid state, whereas monosomy 7 lose heterozygosity throughout the 7p and 7q arm and show drop in ploidy for chromosome 7 SNVs. Our first question for SAMD9-9L patients was the co-occurrence and order of acquisition of those mutations. We found that all SAMD9-9L patients have branching trajectories, meaning that several clones occurred together and only one patient showed a linear trajectory of evolution of monosomy 7. In one representative example with germline SAMD9L mutation showing both branching and linear trajectories, in this patient, we have sequenced or genotyped over 1,600 bone marrow cells. The gray circle on the left represents the native hematopoiesis, that is containing the germline SAMD9L mutation. The somatic compensatory SAMD9L mutation is indicated in blue. Next to it, you will see the UPD7q clone in green in 8% of cells. And finally, on the right are 2 clones with monosomy 7. One has monosomy 7 only in pink, and the other one had additional leukemic ECH2 mutation in 30% of cells, shown in violet. This represents clonal progression towards the leukemic state. In all analyzed SAMD9L patient, clonal trajectories were functionally redundant, aimed to lose the germline mutation, and monosomy 7 is always non-redundant, meaning that chromosome 7 with germline SAMD9-9L mutation is always lost. In contrary, clonal trajectories in GATA2 patient is very diversified, directed to leukemic progression, and monosomy 7 can be random with divergent clonal evolution patterns. This GATA2 case here undergo branching evolution to develop 2 different monosomy 7 clones, where clone 1 is upon losing the red chromosome 7 and clone 2 is upon losing the blue chromosome 7. The clone 1 then linearly evolves to acquire STAG2 inactivating mutation shown in green, and clone 2 acquires KRAS activating mutation in red. The germline GATA2 only hematopoiesis is shown in gray. Another example of GATA2 clonal trajectories is the coexisting monosomy 7 and normal karyotype-driven clonal hematopoiesis. The germline GATA2 patient acquires a known hotspot asexual mutation in blue and develops a separate monosomy 7 clone with ECH2 set domain inactivating mutation and a rare ETV6 mutation shown in orange. This ETV6 variant was shown to contribute to clonal progression in an MDS case that is followed for 18 years in a fantastic case report recently published in Hemisphere. In this last clonal trajectory slide, I want to highlight the extensive branching and linear oncogenic clonal evolution that can be observed in GATA2, something that we do not see in SAMD9-9L patient. This patient here has pathogenic synonymous GATA2 mutation that linearly evolves to acquire monosomy 7 with ASXL1. This monosomy 7 ASXL1 clone then branches to two independent clones, where one acquires KMT2C set domain inactivating mutation, commonly observed in AML but rare in MDS, and the other clone acquires the activating KRAS mutation. For clone 2.1, monosomy 7 caused loss of one copy of KMT2C. The additional frame shift truncation mutation together results in biallelic loss of KMT2C. This clone further evolves to acquire ECH2 set domain inactivating mutation. And also this ECH2 mutation was missed by bulk sequencing and identified only on single cell, emphasizing the sensitivity of this technique. Together, these clones contribute to the GATA2 patient hematopoiesis. This brings me to the last or the end of the talk. Altogether, there are few take-home messages from this study. Firstly, the extreme hematopoietic plasticity of pediatric MDS that contributes to clinical progression, and detection of them will be essential for treatment management. Second, the nature of clonal event can differ by germline predisposition. Some denied 9L patient clonal trajectories are pointed to lose the toxic germline mutation. Hence, patients undergo multiple rescue events. Monosomy 7 in these cases is a rescue clone that can still progress to acquire additional mutation, but the progression is slow and less aggressive than GATA2 patients. In GATA2 patients, all clonal trajectories attribute to leukemic clonal evolution. Lastly, a common theme of pediatric MDS clonal evolution with monosomy 7 is that monosomy 7 is an ancestral clone that undergoes linear or branching trajectories to acquire additional mutation, of which some will expand to acquire more mutational hits, while certain mutations like ASXL1 variants can also disappear. With this, I end my presentation by acknowledging all the people involved in the study, especially my mentor, Marcin Lodarski, my colleagues, Sarni Skutings and Victor Pastor, who contributed to this study. I want to extend my sincere appreciation to Evoke MDS Consortium and Freiburg Study Center for providing me this incredible opportunity to work and learn with pediatric MDS. I also want to acknowledge our funding sources, MDS Foundation for the opportunity to present our study, and a huge appreciation for all primary caretakers, patients, and families. And thank you for your attention.
Due to the lack of time, I think we'll avoid having questions. If you have questions for the speakers, you can contact them afterward. So we'll go on to the second of the four presentations, again, another one by Zoom. And this will be by Dr. Arda Durmaz from the Taussig Cancer Institute at the Cleveland Clinic, abstract number 295, entitled Convergence of Genomics and Transcriptomic Patterns of MDS.
Good afternoon. I want to thank the committee for giving me the opportunity to present my work, and I also want to apologize for not being there in person. So I have nothing to disclose. As a quick intro, as a background, in general, the genomic complexity in MDS or other disorders are underestimated if studied by considering only a unimodal fashion or one feature, either genomic or transcriptomic. Traditional approaches for characterization of disease patterns hence have limitations. The aim of our study is to integrate genomic and transcriptomic data using machine learning as a tool to dissect novel features of MDS. To model this, we hypothesize that splicing dysfunction in MDS may not be restricted to splicing factor mutations only, hence use a data-driven approach to combine multimodal features together. So data integration for statistical learning has been studied from various angles. And relatively recently, dimension reduction has been applied for characterizing low-dimensional manifolds associated with multiple features. And these techniques have been applied in various data sets, ranging from solely clinical to even single-cell multiomics data sets. And these methods range from factor analysis to specific sparse correlation analysis. So with a similar spirit, we aim to integrate multiomic features using a dimension reduction driven strategy. To do this, we chose to use a relatively straightforward and easy to implement tool based on neural networks and autoencoder architecture. So simply, the structure of the shallow network that we created includes a shared layer to capture covariant features across expression, PSI, and mutation, and also individual layers to capture variation unique to individual features. Furthermore, we use variational inference for parameter estimation of the mean and variance of the latent components, effectively characterizing the latent space in a probabilistic fashion. However, note that in order to focus on the covariant features, we utilize only the shared latent layer for downstream analysis, such as clustering, but this is an inherent limitation, but still allows us to filter out patterns unique to single omic types. And subsequently, shared latent layer is clustered using Gaussian mixture modeling. Briefly, the overall workflow of our study includes 3 main steps. Bone marrow samples were collected from approximately 1,200 patients with myeloid neoplasia. 54% of the patients carried at least one mutation in one of the 7 indicated splicing factors, and 90% of the patients carried at least one mutation in a myeloid gene. Copy number data for splicing factors mapping on the chromosomes X, 5, 7, and 17 were also accounted for. A cohort of 63 bone marrow samples from age-matched controls was included. To build a multimodal machine learning tool, we then merged multi-omics data of transcriptomics, genomics. A total of 17,000 approximately alternative splicing events were filtered to be included in the analysis based on the variation in the PSI as well. Finally, a validation model was also applied. We assigned patients based on the latent layer clustering to 15 clusters named C0 to C14. Healthy subjects indicated in black color, which can be discerned from the lower panel, were largely separated from the patients. As expected, patients with splicing factor mutations constituted the extreme pole of dysfunction, which can be delineated from the separation of splicing factors and non-splicing factor cases. However, we show that also patients without splicing factor mutations were characterized by an extent of dysfunction as well. We also noted a small fraction of cases showing patterns similar to healthy controls. Among these, we identified carriers of SRSF2 and SF3B1 mutations with low variant allelic frequencies. Moreover, patients with splicing factor mutations were mostly enriched in MDS clusters. To investigate the composition of identified clusters, we used CyberSort analysis to confirm a similar composition of myeloid cells across samples. As is shown on the upper pie charts, the composition of myeloid cells was uniform across all clusters with a median of 75% across the clusters. This suggested that the identified clusters were not due to different cellular compositions. Similarly, to better understand the dominant leukemia hierarchy represented in the patient cohort, we performed the same analysis using a different signature and malignant signature panel described by Zhang and colleagues. Out of the 7 cell type configurations, all clusters were mainly overrepresented by GMP-like composition, again, suggesting that the identified clusters did not significantly differ in terms of differentiation hierarchy. We then compared the distribution and similarities of all mutations across the 15 clusters. A couple of examples include clusters 1, 3, 7, and 12 being predominantly composed by SF3B1 mutants, whereas clusters 8 and 14 consisted of SRSF2 mutants. Non-splicing factor mutations were equally distributed in different clusters. For example, DNMT3A and TET2 had approximately 0.1 mutation frequency across the majority of the clusters, including clusters 9, 10, and 12. Interestingly, ASXL1 showed increased mutation frequency in clusters 8, 11, 13, 14, and was mutually exclusive with SF3B1 at higher frequency levels. We then investigated the association of splicing factors, mutations, and the PSI levels. Since we built a probabilistic framework, we were able to sample from latent space and capture the linear association. Identify subset of features characterized by similar correlation patterns across mutations and PSI features. For instance, SF3B1 mutations show positive correlation for specific SLC25A37 gene isoform, which is an iron importer and was previously associated with the SF3B1 mutant myeloid neoplasia, with similar association with other splicing factor features like LOX7L2, PRPF8 deletion, and ZRSR2 mutation as well. In contrast, we observed that patients with ZRSR2 deletion, SRSF2, LOX7L2 mutations were characterized by positive correlations with a different set of isoforms associated with EZH2, suggesting that SSF2 mutations might be not exclusive in promoting missplicing of EZH2. Further investigating the mutation PSI associations for cases with TP53 and EDX41 alterations specifically points out possible convergence mechanisms. For instance, samples with TP53 mutations, LUX7L2, PRPF8, and EDX41 deletion showed increased expression of some genes like EIF4H and PDGFRA, FIP1L1 fusion genes in cluster 0, compared to samples with the same mutations in cluster 5. Similarly, a heterogeneous group of samples in cluster 2 with and without TP53 and PRPF8 mutations showed decreased expression of splicing factors and other helicases, SSF5, DDX6, and DHX9 expression. We further investigated gene expression patterns across the two major groups identified by the shared latent space. Differential gene expression testing and subsequent pathway enrichment analysis resulted in immunity-associated genes. We observed the distinct expression patterns of various HLA genes across the two groups, including clusters 9 and 10, which are both almost purely AML clusters, where cluster 10 had decreased expression of various HLA genes, including HLA-B and DRB1 allotypes. And proteasome complex genes were relevant as well. Specifically, downregulation of associated genes suggested possible T-cell surveillance escape mechanisms. Nevertheless, we utilized a relatively simple approach for data integration, which showed relatively high stochasticity of cluster assignments, where in contrast, a vanilla version of the proposed architecture actually improved upon the clustering accuracy based on the adjusted randomness. Furthermore, we observed a significant portion of variance explained for the expression data by the individual layer, suggesting that utilizing a combined latent space or a more deeper architecture or more sensitive architecture might be better effective for downstream clustering tasks. With that, I want to thank all our collaborators and funding sources, specifically Dr. Valeria Visconti and Dr. Yaroslav Maciejewski, and I want to thank you all.
Thank you very much. And we'll move on to the third presentation.
So the next presentation will be presented by Pedro Moura about accelerated RNA splicing dynamics during erythroid differentiation, which amplifies missplicing in SF3B1 mutant MDS-RS. Pedro is a postdoc at Karolinska Institute in Stockholm, Sweden.
Thank you for the introduction, and hi everyone. On Wednesday I presented a little bit more clinical. Today it's a little bit more molecular, but first I said I have nothing to disclose. So I'll get right on the background so we're on the same page. So I wanted to just focus a little bit on the pathobiology of MDS-RS. As you all know, it's mostly caused by mutation in the SF3B1 gene. Part of that blob you see there is the human spliceosome. And when a mutation happens at the hematopoietic stem cell level, it leads to RNA missplicing of many genes, but in the pathobiology that leads to the creation of ring sideroblasts, there's been a decade and a half of very detailed studies that identified specific missplicing of these three genes, ABCB7, PPOX, and MAP3K7, as essential in causing nonsense-mediated decay, which implies that the missplicing event that has affected these genes introduces a nonsense codon, which in turn leads to degradation of the transcript. That leads to downstream iron dysregulation, which then affects the erythroid lineage, most importantly, causing anemia. And the cell you see there is the ring sideroblast, and thus it's the central feature of MDS-RS. Now, despite being the central feature of MDS-RS, and we know very much about how the ring sideroblast is generated, we know depressingly little about what the ring sideroblast does, how it maintains its existence, and whether anything happens after it is formed. And so that was our curiosity during this project. What happens when the ring sideroblast is formed, and is there any point at which SF3B1 mutation affects cell biology that we don't know about? So we started kind of basic. We went to mononuclear cells of MDS-RS patients, and we looked at a classical way to assess erythroid biology, glycophorin A, and then sorting into band 3 and integrin alpha 4 subsets. And we observed what was presented by Professor Fontenay in the culture system as well, that the patients appear to have an increase in the progenitor subsets, but a relative decrease in the most differentiated orthochromatic erythroblast subsets later on. We looked at cell cycle and Ki67 analysis to assess whether the cells maintain cell cycle activity. Here we could not identify any differences between normal and MDSRS patients. When we looked into the ring sideroblast compartment by sorting cells from these subsets and then performing Prussian blue staining, we observed that there was an enrichment of ring sideroblasts the further in erythroid differentiation the cells continued, but that this in fact only leads to a normalization of the erythroid biology in the MDSRS of the ring sideroblast population, and that there was also a decrease in the cycling of the ring sideroblasts, but nonetheless that the median ring sideroblast frequency was the same in the cells with the median Ki67 frequency. Then we assessed the function of these cells through 4-week erythroid culture, and importantly at this point we observed that the enucleation frequency of the ring sideroblast was exactly identical to the enucleation frequency of normal blasts from the same patients, meaning that now we have established these cells have the capacity to differentiate, to divide, and to enucleate, replicating normal erythroid maturation. And at this point, we came into a problem, which was that there was no way to really look into the ring sideroblasts specifically. So we thought hard, and we took a page from another side of erythroid pathobiology, which in malaria for the same decade and a half, people have used magnetic separation to extract malaria-infected cells. And we tested passing both mononuclear cell layers and the high-density red cell layer through a magnetic separation column. And in fact, we happily report that by doing this, and we can then further purify the ring sideroblasts and the siderocytes through FACS sorting if extracted from the red cell layer. We obtain a highly significant enrichment from either cell source, but we obtain many more cells from the high-density layer rather than a mononuclear cell layer. We report as well that by doing this and assessing the genetic makeup of the ring sideroblasts, that this population is entirely clonal, which is not a surprising finding, but it's still the first time that this has been described. And given the ease of isolating these cells just by magnetic extraction without any specific marker, we then wondered, are we missing anything? Because CD34 magnetic extraction is the core technique in the field. It's been used in many of the previous studies defining MDSRS and its contribution in the stem cell compartment. And indeed, we find that CD34 max separation leads to the non-specific enrichment of a glycophorin A positive, a CD34 negative. Ring sideroblasts do not have CD34 positivity at the surface, but they are specifically enriched by the magnetic separation procedure, and we don't see that in the MDS without RS, meaning that this has some implications to how we assess the transcriptomic data of MDSRS patients. So armed with the ability to look into these cells, we performed a comprehensive multi-omic analysis. We performed bulk RNA sequencing to be able to look into the missplicing specifically. We performed single-cell RNA sequencing to look closer into gene expression, as well as confirm our missplicing observations. And then we finalized this with proteomics of the ring sideroblast and erythroid compartments and really look at the holistic view of what is and how does a ring sideroblast function. I'm not going to spend much time in the gene expression other than to say that we now have defined everything that the ring sideroblast does in order to survive. It activates several pro-homeostatic pathways, which if you look closely enough, you recognize a few of them, but mostly addressing the missplicing and oxidative stress that is predominant in these cells. And I'll also mention the top hit in all of the data sets, which was GDF15. I believe also mentioned a couple of times previously in this meeting. And we observed the specific expression of GDF15 by the ring sideroblast population at the RNA level and at the protein level and secretion level. And then we assessed, is the GDF15, could that be doing anything to the hematopoietic stem and progenitor cells? And we identified a small but nonetheless significant effect on wild-type cells only upon adding GDF15 that leads to reduction in colony formation ability. Now I want to focus on the main part of my talk, which is RNA missplicing and the central feature of SF3B1 mutation. We observed that by looking into the ring sideroblast and comparing also the siderocyte, which has remnant RNA, but nonetheless descends from the ring sideroblast and allows us to look into erythroid missplicing, that missplicing is a highly cell-type specific event. So the number of genes that are statistically significantly misspliced between the HSPC compartment and the erythroid compartment is actually very small, as you can see by the intersection of these two groups. And the genes that go into either of these groups independently are cell-type specific. So in the hematopoietic stem cell compartment, we have many more stem cell-related genes, and vice versa in the erythroid, we have erythroid genes. But if we look into the type of missplicing that's also happening, we see a change. In the CD34 compartment, we observe what has been reported by several before, that there is missplicing specifically with intron retention and alternative 3' splicing, which changes progressively throughout erythroid differentiation and then remains the same after enucleation into exon skipping events, which seems to be a very specific feature at the ring sideroblast level. We then looked into several previously validated genes, and we observed that the missplicing level already increases in the glycophorin A positive. This is mixed erythroblast population, and these have the same variant allele frequency as the CD34 population. And then it increases further in the ring sideroblast population. And here we know that the ring sideroblast population is fully clonal, so we thought there could be potentially an effect. But if you look at the SF3B1 missplicing event, you see that this is clearly much more than linear, and it's a highly significant effect. So then we started thinking of ontological reasons for why this could be the case. And it is well known that erythropoiesis is an hemoglobin-producing machine. Cells arriving at the end of erythropoiesis are basically full of hemoglobin RNA and ribosomal RNA to deal with that, and you can see this reflected. This is from 10x data of 5 MDS patients and 3 normal bone marrow controls. There is no difference between normal and MDS. And the percentage of spliced transcripts increases with progression through erythroid differentiation, but this includes all transcripts. We then decided to remove the hemoglobin and ribosomal transcripts and assess all other RNA. And in fact, we see the same thing. So RNA splicing accelerates already at the early erythroblast level, far before the cell has the ability to stop transcription. In fact, it's extremely active at this point, and still it accelerates, and this is maintained throughout differentiation. So we think there is an effect here that also, together with other molecular effects, leads to the prevalence in the exon skipping event that we are seeing. This was a completely computational prediction, so we wanted to add a little bit more ground truth. So we boosted our 10x data with SmartSeq3 data, in which we sorted the progenitor populations as well as the erythroblast populations. And then we assessed exactly the same metric, the percentage of spliced RNA reads. And we happily confirmed that indeed the erythroblast, and here it's looking only at RNA without the hemoglobin and ribosomal transcripts, do have an accelerated RNA splicing phenotype. What are the consequences of this? Well, here I focused only on known targets, but together with the proteomic data, we were able to identify that because there is a loss of ability to undergo non-disease-mediated decay and accelerated ability of the splicing, then we see that the RNA splicing begins to be corrected through overexpression rather than being removed entirely. And so we see that for several SF3B1 targets, you can see ABCB7 there is the main target, but for several others where RNA overexpression cannot compensate the protein expression loss in the cell. And so we expanded this idea into further analysis, into a more general analysis, because in the bottom left corner, we're looking into known targets. So what happens if now we look at highly expressed RNAs, but don't lead to a sufficient increase in protein or the same relative increase? So here, the scale in the RNA level starts at 2 log-fold change, which is a 4 times overexpression, and the scale in the protein stops at 0.5 log-fold change in non-significant increase, and from there to below. And the only thing that we found to be significantly enriched in terms of pathway analysis was pro-apoptotic genes, for which you see several here, which are related to the TP53 pathway. To our knowledge, this is the first time this has been identified in MDS-RS with SF3B1 mutation. And the proof of the missplicing pudding is in the sashimi plots, which, if you've never seen one before, this is showing the exon, intron, exon junctions. And you can see that both for FAS and BAX, two of the targets we identified in this, we observe the insertion of new exons, which are known in the literature to normally induce nonsense-mediated decay. But here we have an accumulation of this, meaning that the few protein that remains in the cells is likely non-functional and truncated at the point where it should conventionally just be lost at the transcript level. And so this leads me to my conclusions. We have performed a multi-omic investigation of the ring sideroblasts, defining this cell as unexpectedly active transcriptomically and cellularly. We show that RS maintain erythroid differentiation potential, including enucleation. We show that these cells engage several mechanisms to deal with the missplicing and oxidative stress and identify GDF15 overproduction as a detrimental effect to the wild-type HSPC compartment. We identified that SF31 RNA missplicing is enhanced by erythroid differentiation in terms of number, frequency, and variety of the misspliced transcripts, and we identify missplicing of pro-apoptotic genes, which directly link SF31 mutant missplicing to TP53 pathway dysregulation. So I would like to thank this talk by thanking everyone, but especially Eva, who's a wonderful mentor. I thank the committee for selecting this abstract and giving me the opportunity to present here, and I thank all of you for listening.
Thank you very much. And the last speaker is Celine Philippe, who is doing her postdoc at Bart's Cancer Institute in London. And she will, the title of her presentation is Targeting the Defective Pathway to Improve Erythropoiesis in SF3B1 Mutant MDS RS Patients.
Thank you very much for the introduction. So good afternoon, everyone. First of all, I would like to thank the MDS Foundation Committee for selecting my abstract for the award, and also for giving me the opportunity to present my work today. So this project that I would like to present is about targeting the defective CoA pathway in order to improve erythropoiesis in SF3B1 mutant MDS-RS. I have nothing to disclose. So as you know, MDS with ring sideroblasts is associated with a high prevalence of SF3B1 and defect in erythropoiesis. And now SF3B1 mutant MDS is recognized as a distinct nosologic entity. So mutation in SF3B1 alters the recognition of the 3' splice sites, and as a consequence, there can be a deletion or addition of nucleotides in the 3' of the intron. As a result, this generates aberrant transcripts that can encode for aberrant proteins, or if a pretermination codon is created, in that case, the transcript can be degraded by the nonsense-mediated decay. So mutation in splicing factors impact hundreds of transcripts that are involved in plenty of different mechanisms, such as proliferation, metabolism, or even epigenetic. And it makes it very difficult to pinpoint which missplicings are actually driving the disease. So historically, splicing analysis has been performed on CD34 positive cells or mononuclear cells. But because MDS is characterized by a differentiation blockage, we thought about performing our experiment on progenitors derived from coloning-forming assay that have been grown upon normoxia or hypoxia. And for this splicing analysis, we pulled all the colony together, so BFUE, G, M, and GM. And so we used samples from 3 different healthy donors and 3 MDS SF3B1 mutant patients. So with this analysis, we capture more than 2,000 missplicing events. And among the most affected pathway, we found the heme metabolism and the TCA cycle to be downregulated. And we wanted to find a target that can explain this phenotype. So we cross-referenced the data that we got in our analysis, and we found a list of 200 missplicing that were found in patient samples upon hypoxia and normoxia, and they were not found in the healthy samples. In this list, we validate missplicing that has been found in the literature. And we also highlighted new targets, and we actually got very excited about COSI mRNA, which was misspliced as high as 40% in our data set. and COSY is actually tightly linked to heme metabolism and TCA cycle. So COSY is a coenzyme A synthase, and mutation in COSY leads to a disease called neurodegeneration with brain iron accumulation. And actually, it's very interesting because this disease shares some phenotype with MDS, such as a cellular iron imbalance and heme biosynthesis defects. So the missplicing in COSY is actually located in the 5' UTR of the transcript. So we perform a PCR on this region in order to validate this missplicing. And as you can see on this PCR performed on healthy donors and patient samples, we got isoform switch. And interestingly, this isoform highlighted in yellow here is actually looks more specific of the patients. So it's actually encoding for a protein called COSY beta. And if we quantify by RT-qPCR this isoform in a bigger cohort of 24 patients, we can find here a really significant upregulation in SF3B1 mutant samples compared to SF3B1 wild type and healthy donor samples. So we hypothesize that this missplicing leads to loss of COSY protein, and because COSY encodes for the coenzyme A synthase, what we expect to see is also a depletion in CoA. And actually, CoA is feeding the TCA cycle, so we also expect to see alteration in the TCA cycle. And to go further, the TCA cycle actually provides the metabolites necessary for the heme biosynthesis, so we also expect from that some defects in erythropoiesis. So in order to carry out further functional analysis, we first validate those results in cell lines Y type or mutant for SF3B1. And here, so we have some K562 that we edited by CRISPR-Cas9 in order to insert the mutation K700E. So we got two clones, C1 and C2. And we also have the HNC34, which is a cell line derived from a patient with the mutation K700E. So as you can see here, we actually recapitulate the profiles that we have seen in patients. And if we look at the protein level now by Western blot, you can see a clear downregulation of COSY at the protein level. And this is also something that we validated on patient samples. So here on CD34 positive cells, you can see a downregulation of COSY protein in the mutant SF3B1 compared to the Y-type SF3B1 patients. So we have missplicing COSY associated with a loss of protein. So does it impact the CoA synthesis and TCA cycle? So here the CoA synthesis starts with vitamin B5, which is transformed into phosphopantothenate, and actually COSY processes the two last steps of the CoA synthesis. And this CoA then feeds the TCA cycle that provides a succinic CoA, and along with glycine, succinic CoA are actually the metabolites that initiate the heme biosynthesis. So we perform a CMS analysis in the cells, Y-type or mutant for SF3B1. And as you can see here, so upstream of COSY, we have a slight increase in phosphopantothenate, while we can see actually downstream of COSY a depletion in CoA and succinic CoA. And also, very interestingly, we could see a depletion in glycine. So then we know that it's not a complete loss of the protein. We have still 40% of COSY protein in the mutant cells. So we wonder if we provide more vitamin B5 to the cells, can we boost the production of CoA? So we actually treat the cells with labeled vitamin B5 at a medium equivalent concentration. And if we have really an alteration in COSY in our cells, what we should see with this experiment, it's even greater accumulation of the phosphopantothenate upstream of COSY. And this is what we saw in our analysis. While downstream of COSY, we can see a rescue of the CoA and succinyl CoA. And as expected, glycine is not affected because glycine comes actually from an independent pathway. So it comes from the serine biosynthesis pathway. So very briefly, so the glycolysis gives first a 3-phosphoglycerate that is converted by the enzyme PAGDH into 3-phosphopyruvate. And then this pathway finally gives serine that is finally converted into glycine. So it was interesting to see glycine down because actually what we know from the literature is that the transcript that encodes the enzyme PAGDH is misplaced in SHB1 mutant samples, and this misplacing creates a pretermination codon, and so finally there is a degradation of the transcript by the nonsense-mediated decay and a loss of protein. So what we saw in our analysis is that the glycine was down in the mutant cells, and we checked also the serine in our analysis, and we found also a downregulation of serine. And also we corroborate the results that were found previously. So you can see on this Western blot that we also got a loss of PAGDH protein in the mutant cells. So this was very interesting, but it was already published, so we went back to the COSI misplicing that were not described yet. And we wondered actually what was the role of COSI in normal hematopoiesis and also in the MDS phenotype. So we took advantage of a model which was really well established in the lab. So we selected some CD34 positive cells from umbilical cord blood that we transduced with the shRNA that target COSI. And we sorted the cells on CD34 and GFP. With those cells, we performed some colony-forming assay, and we could see first a reduction in the total number of colonies that can be explained by a reduction in BFUE and GM and GM. So then we perform a erythroid differentiation in liquid culture, and we follow the acquisition of the CD71 and CD235A. So to go through briefly, so the cells first acquire the CD71, then the CD235A, and finally the cells are losing the CD71. So if we focus on this quadrant number 4, where are the most mature erythroid cells, what we can see is that the SH-COSI actually affects this category. So we have less mature erythroid cells with the SH. And also, interestingly, if we check quadrant number 3, so the cells are less mature. And here we can see actually an accumulation of the cells in this compartment with SH-COSI. And this is actually a phenotype that really resembles to MDSRS. So I've shown you that COSI is important for the generation of succinate-CoA. It's very important for the synthesis of heme. And we also check the heme content in the cells. And with SH-COSI, we could actually see a reduction in heme production. So finally, we try to see, so with those findings, if we can use it to improve erythroid differentiation in MDSRS patients with patient samples with SHB1 mutation. So we selected the CD34 positive cells from those patients and pushed the cells towards the erythroid differentiation. And we treat the cells with vitamin B5 or succinate-CoA. And then we follow the phenotype, so CD71 and CD235A. So what we can see on those 4 patients, so we can see some heterogeneity, of course, but what we can see also is they are at least sensitive to one of the treatments, so we improve the erythroid differentiation with at least one of them. And if we pull all the data together, we can see that actually we got a significant improvement of the erythroid differentiation cells in the primary samples with both treatments, vitamin B5 and succinate-CoA. Finally, we measure the heme, and what we can see is that with the succinate-CoA, we can actually improve the heme production. So to conclude, I'll show you that SFCB1 mutation misplaces COSI mRNA, and this induces a loss of protein. As a consequence, we have a depletion in CoA and succinate-CoA. And what we can see also is a heme synthesis impairment as a result of that. And this heme impairment, of course, participates to the erythroid differentiation blockage. And what was really exciting is that actually we could play on that and improve the erythroid differentiation by treating primary cells with vitamin B5 or succinate way. So I'm very glad to say that we published this story in Science Translation and Medicine 1 month ago. So with that, I would like to thank everyone who has been contributed to this project, and especially Kevin and Said. If there are students in the audience who are interested by what we are doing, please feel free to contact Kevin. And thank you very much for your attention.
So that concludes the scientific program for today, and we'll now have the closing remarks.
Yes, I will ask Ariane to join us. So thank you very much to all of you for being present. Before that, we have to give the prize to the 2 awardees that are present here. So please join us. Okay, so I will start a long speech. You ready? No, I try to be very brief. So I just wanted to also thank all the speakers who've done a very great job. I think that we've all get a nice overview on the current knowledge on myelodysplastic syndrome, and it was important. In particular, we had all this discussion regarding the classification, and it will be important for all of us to help us using this classification and stratification systems in our clinical practice. I was also struck by the importance of the research on the description and deciphering of the early steps of the disease, which opens the way for new therapeutic strategies, for interception strategies, which might be a real change in paradigm. And of course, we've seen some interesting preclinical data indicating that we can be optimistic for the future, and that we will probably have interesting future new agents. And before I finish, I just want also to thank again my co-chair, Pierre Fenot, who had to leave, but who has done a tremendous job for the organization of the meeting and for the scientific program. I would like to thank the sponsors, the MDS Foundation, KENES, and all the local organizing committee. So now I'm passing the floor to Ariane, and I'm also passing not the Olympic flame, but the myelodysplastic flame. Okay, go ahead.
So, at the end of this meeting, I first would like to congratulate you, Norbert and Pierre, and the scientific committee members, as well as the MDS Foundation, for this excellent symposium here in Marseille. So, to continue then, I would like to speak on behalf of my co-chairs for over 2 years, Saskia Langemeyer, who's also in the audience, as well as Theo de Witte, to invite you to come to Rotterdam and save the date already in the beginning of May in 2025. And why Rotterdam? I think Amsterdam is even better, but I would like also to invite you to Rotterdam because Rotterdam is a thriving cosmopolitan city. It's the gateway to Europe. It's one of Europe's greatest ports. It's an excellent access by flights, by rail, over water, or whatever you would like to come over. It is a top quality international renewed knowledge in institutions as well as in our universities. It's a highly budget-friendly city and a rich architectural heritage and design all over the places in central Rotterdam. And Rotterdam is a young, trendy and dynamic city. So I would like to invite you, and already on behalf of my co-chairs, Saskia Langemeyer and Theo de Witte, I'm lovely to share the scientific committee together with members in the Netherlands, which I will name already, Marissa Westers, who's in my group, Janan Alan, who's also in my group, but also Joop Jansen from Nijmegen, Dr. Gerwin Huls from Groningen University, as well as Mark Rijmakers, who will be part of the scientific committee together with the MDS Foundation. So why also the Netherlands? Because dependent on how you look to the Earth, it is the central of the world. And you can easily come over by airplanes, because over 300 of flights coming daily, several times from all over the world, all continents, centrally in Amsterdam. And also close to Rotterdam, there's a smaller airport, the Hague Rotterdam Airport, and within Europe, over 40 destinations on a daily basis. But from airport Schiphol International, you can easily go by high-speed train only 25 minutes every 4 to 6 times per hour to come from the airport. Just 5 minutes walk after the customs, you go to the platforms, and you are easy going by train to Rotterdam. And also high-speed trains from France, from Germany, and for other countries within Europe come centralized in Rotterdam or Utrecht or Amsterdam. So quite convenient to come over. So finally, I show you a small film, only a couple of minutes, to convince you that Rotterdam is the place to be in the over 2 years for the 18th MDS Foundation, MDS International Conference. So please look to the film, and I hope you have a nice travel back home. Thank you for your attention.
A city with a passionately beating heart.
We want to change the future.
I see a lot of great innovation happening in Rotterdam. The world's largest port. Boundless ambition.
It's truly the connections that people make here. We're growing the next generation of world-changing businesses.
So, welcome in 2 years. Thank you.